We're going to get started in about 1 minute. We're just giving time for all the participants to be able to join into the webinar. So just bear with us for about another 30 seconds as people are able to connect. Thanks so much. Susan, I think we can go now.
Okay, sure. Welcome to our first session on discussing gene therapy. This session is titled Developing and Designing a Gene Therapy Program. Today we'll be discussing proof of concepts that lead to a program from initiation through validation. Our goal in hosting this session is to provide an opportunity to hear from experienced researchers and clinicians in the industry. Thank you for the questions that have already been submitted. We have incorporated them into our Q&A. Jen will be monitoring the questions submitted during the presentation. You can submit those questions at the bottom of your screen in the Q&A chat box. We will include as many as possible during the presentation and also at the end. But if at the end of the session, there is an email that will be provided where you can continue to send in your questions if you still have additional answers that need to be addressed. Our speakers today are Dr. Michael Binks, who is the VP of Rare Disease Clinical Research at Pfizer, and Gabby Pretzel, Director of Neuroscience External Research at Takeda Pharmaceuticals. We are grateful for your time and expertise to educate our FA community, so welcome and thank you. Before we get started, I just want to reiterate our aim. So the aim of our discussion today is to cover high-level principles regarding development of gene therapy programs. During this discussion, the panelists will not discuss timelines of when gene therapy clinical trials are expected to be available for FA, and we won't be discussing specific details about any program in development, including expectations for eligibility criteria and data collection. The idea is to give you ideas of program development that will probably apply to an FA program. So let's first review the principles behind designing a gene therapy program. Michael, can you tell us a little bit about the diseases that are amenable to gene therapy?
Sure, Susan. Gene therapy is not a new idea. It's been around for many decades, 40 or 50 years probably, really since we got a good understanding of our ability to clone genes and eventually sequence the entire human genome. But clearly in inherited disease where there's a single gene that seems to be causing the disease, as in the case of FA, the mechanism of disease can be very well understood, and in some instances, being able to replace the gene or the gene product in such a way as to allow that gene to function properly is a very attractive and tractable, viable way of dealing with a disease. So there's been a huge amount of activity over the last 20 or 30 years to find ways to introduce a faulty gene or a deficient gene into the right cell type and tissue that could perhaps ameliorate the disease and reduce dependency on other treatments, and perhaps get to a point where the disease plays a much lesser part in people's quality of life.
Mm-hmm. And so the examples on this slide, are they examples of active gene therapy programs?
Well, among cystic fibrosis is, as you know, probably the commonest genetic disease, and gene therapy programs started way back in the late 1980s and 1990s with mixed success. And you know, the field has been continued working in that area, and you know, there are still some promising leads in cystic fibrosis. In hemophilia, which is a genetic deficiency of clotting factors, there are now multiple programs in advanced clinical development, and we hope that there will be approved treatments for hemophilia based on gene therapy in the next few years. Likewise, muscular dystrophy. Actually, I've been the project leader for a gene therapy program in Duchenne muscular dystrophy. The gene was identified back in the 1980s. It took 15 years to identify technology that could be used to deliver a gene that was small enough to package into a gene therapy vector. And now there are multiple clinical programs advancing gene therapies in Duchenne muscular dystrophy. Sickle cell disease is a little bit different, where it's possible to take cells out of the bone marrow and introduce a gene therapy essentially in a test tube, and then reintroduce the modified cells back into the circulation. And that's proving to be a very exciting new therapy for sickle cell disease. And eye diseases are on the list here because they've been a relatively easy way to introduce this kind of technology, because the eye is a small confined space, and so you don't necessarily need very much of the gene therapy drug to deliver there. And also the eye is a protected space from the immune system. So there are some advantages to delivering gene therapies there. And so now in 2020, there are several approved gene therapies based on viral vectors that have been approved for spinal muscular atrophy, a severe musculoskeletal disease that is the cause of the most common cause of the most common genetic disease causing death in infants, essentially. And one eye disease, which has responded very dramatically to gene therapy. And there are a number of others in development.
So you mentioned viruses, viral, you've mentioned that word several times. Can you tell us more about the components that go into a gene therapy?
Sure. So viruses have evolved to be able to get inside of cells and hijack the machinery of cells to make their own proteins. Now, some of those abilities we can use to deliver a therapeutic gene. And there are a couple of families of viruses that appear to have been particularly successful. And if you look across all of the gene therapies in development, by far the majority use what's called the adeno-associated virus family. And these have been selected partly because they're particularly good at getting into cells and getting genes expressed in cells, but also because they have a fairly low level of, they cause a very low level of inflammation and relatively little immune response compared with other viral families. So what we do from the virus is remove the genome and use parts of the genome and introduce a therapeutic gene under the control of what we call a promoter that drives the translation of the therapeutic the transcription of the therapeutic gene and results in the production of the protein. But first of all, it has to get into the right cell in order to deliver the gene to the right place.
So it sounds like what you're saying is that the AAV is no longer infectious in terms of what we think of in viral infections, but it's essentially just carrying, we're engineering it to carry the gene of interest only.
Correct. These viruses cannot replicate themselves.
Got it.
And that's very important that from a safety perspective, even though the AAVs don't really cause much in the way of disease as a natural virus.
Right, seems like a perfect use. So going from this concept, how does the virus vector know which tissue to go to? So,
of course, it doesn't know, but it's clear that viruses have evolved in many different ways, and there are many variants of just this one family of viruses, the AAVs, that have slightly different characteristics. The proteins on the surface are shaped in slightly different ways, and it turns out that some of those differences in shape of the surface of the virus confer preferential uptake into certain tissues. So the field has learned over the last decade or two that different variants of the virus have propensity to be able to get into the brain specifically, or to muscle or the eye, or into the heart, the various different tissues. Now these aren't pure specificities, they're relative differences in uptake. But clearly, if we're able to deliver the virus preferentially to one tissue over another, that can have a big advantage in terms of the dose that's required, and also perhaps have an effect on the safety. But it's not quite as simple as it seems. This was originally, mapped out in small animals, in mice in particular. These affinities for different tissues don't always track when you move from one species to the next. If you move from a mouse to a dog to a monkey to man, they're not necessarily always the same. So we've got to be a little careful about extrapolating from small animals.
That makes sense. And that, yeah, you're right. This slide seems fairly simple, straightforward. Pick one off the shelf and you're good to go. So it's good to understand that perspective of understanding different animal models in which this is tested. And I'll come back to that towards the end to pick up on that point again. But I appreciate your perspective that it looks simple on paper and it can be much more difficult in practice.
And one other thing to say is that, although many of these emerged through evolution, it's been possible to engineer smaller changes that can have subtle distinctive differences in the properties of the vector. So there's a huge amount of innovation in this space, trying to identify variants that are particularly good, even better than the naturally evolved genotypes or structures.
And so I would imagine that there's, so what I'm getting from that is that there are companies that maybe just work on the viral capsid construct, but don't necessarily have a specific gene therapy program. And then would certain companies collaborate to possibly use a re-engineered virus for a particular program in-house?
For sure. There's as I say, there's a huge amount of activity and some very large libraries of capsids that have been screened for different properties.
Great. Okay, so now I'm going to bring it down to a patient level. And when a patient is given a gene therapy, what is happening at the level of the patient's cells?
Right. So again, thanks for the question. So the key thing that has to happen is uptake into the cytoplasm of the cell, and this happens through a process called endocytosis, where the cell membrane wraps around the vector and then delivers it into the cytoplasm, where it releases the nuclear, the genetic component of the vector into the nucleus. And there it can start doing its work as a gene. But this is a very complex process that we partly understand the uptake. Some of it is non-specific, and but there are clearly tissue-specific elements to the interaction between the cell membrane and the vector itself. But it really has to get inside the cell. The vector, the capsid needs to release the nuclear material, and it has to get into the nucleus. And all these parts of the steps have to function well in order to get the expression of the protein that you're trying to get expressed.
So I would imagine that not every virus is taken up into a cell. So any virus that is remaining circulating in somebody's system, is that what is generating the immune response?
Well, certainly the immune response to the virus is generated partly from circulating vector.
Okay.
But not only so. So the proteins that enter the cell can present themselves to the immune system in such a way that the cell is directly attacked. So the components of the immune system are complex, but antibodies form, which are the proteins that bind to the surface of the capsid and will extract them from the system. They'll be cleared out through complex cleaning systems, if you like, in the liver and spleen. But in addition to that, the capsid proteins can make themselves available for other immune cells to directly attack the cell that's been transduced.
Got it. I know that it's often a question from our FA community in knowing that FA is a multi-system disease. And we can talk about FA in a few minutes, but what are some modes of delivery for gene therapy that have been used in programs so far?
So before we get there, maybe we should just address this this slide, which raises the phrase episomal. And so for this family of viruses, the AAV family, the genome almost always, as far as we know, sits outside of the host's chromosomal DNA in a separate structure that is transcribed separately. There are other types of viruses, viral genomes and viral vectors that cause the integration of genes into the host DNA. And I think one of the reasons AAV has been popular as a viral vector is that this DNA always sits outside of the host chromosomes. And therefore the risk of introducing an unexpected mutation or something like that is very, very much reduced. So hence its popularity. But an episome is a bit of genetic material that sits outside of the chromosomes, and I think it's important to introduce that term.
Thank
so,
you.
but you're going.
Sorry, Michael, to interrupt. We just had one question from the audience that goes back just a little bit when we had the picture up of all the different AAV capsids and how they target different tissues. And when we were talking about how we make it look simple right here, somebody's asking, do the AAVs that target the brain also target the rest of the nervous system? And I guess I take that maybe even one step further and say, you know, when you think about the brain and neurons, there are lots of different types of neurons even in the brain, and the brain itself is a large organ with lots of different regions. And so maybe you could kind of speak to what we mean by brain here.
Well, there are certainly many different cell types in the brain. You know, there are neuronal cell types, and there are microglial cell types that clean things up, and many others that that are subspecialized in one form or another. And we're really just learning now about the affinity of particular capsids to different different cellular types within the brain. But for sure, there are cellular specificities within the brain. And, you know, that adds to the complexity of choosing the right vector to deliver to, well, as Gabrielle will talk about, the numerous different cell types that maybe you need to target for a disease like FA. Does that answer your question?
Yes,
Jim?
thank you.
So
we had started talking about administration. Do you want to go there, or did you have?
Sure. No, let's do that. If we can go back one, or is it forward one? Yeah.
This one? Sure.
Yes, I think it's important that to appreciate that these gene therapies in different diseases are necessarily delivered in different ways. So if we want to deliver to the liver, deliver the vector to the liver, we'll generally deliver it intravenously. Or if we want to get into muscle, we would deliver it into a vein in the arm. Or if we want to get into the heart, we might deliver it through a vein in the arm. But when you need to get into the central nervous system, it gets a bit more complex. It may be possible to deliver it through a vein in the arm, but equally, that might require us to give so much of the vector that we get lots of side effects. So it's important to understand that there are alternative routes where we might be able to deliver much more locally, either into the cerebrospinal fluid through essentially a lumbar puncture, or into the brain itself. There are other routes into the cerebrospinal fluid. So if we flip to the next slide, or is it the one before? It's that one. Yeah. You can deliver into the cerebral ventricles, which are fluid filled, to distribute the vector around the tissues adjacent, or there is a route in close to the brainstem called the cisterna magna that can allow us to get into the CSF near the brainstem. The other alternative is direct injection into the cerebral or other CNS tissue. So not into the fluid so much that surrounds the brain, but into the tissue itself. And all of these are options as a route into to get expression in appropriate regions of the brain relevant to that disease state.
It also sounds like, from what you were saying, not only is it getting more specific to the tissue of target, but by directing it to that tissue directly as much as possible, it decreases the risk of side effects that may be very unwanted in that patient.
It certainly may do that, and it may reduce the amount of vector that you need to get expression in the relevant tissue. Absolutely. And these are complex experiments, again, different between species in terms of these routes of administration. I don't think anybody's cracked it from the point of view that there is no approved gene therapy for delivery to the CNS directly.
Okay. So I'm going to switch gears into talking about FA specifically, but Jen, are there any questions that we want to address on this section?
We do have a few questions that have come up, but maybe I'll hold them for just a few more minutes, because I think they're great questions, but they're going to tie a lot of things together for us.
Great. Okay. Gabby, I'm going to ask you to talk a little bit about why FA is a viable candidate for gene therapy. So if you want to do a brief review of FA, just to give us an idea of.
Sure. Yes. Yeah, sure. And thank you for having me on this panel. This is excellent. So as it started out with the first slide, what Michael reviewed is that monogenic diseases are, the really perfect for gene therapy, and then in addition, loss of function, because we know the cause of the disease, and we do know, and we have to just basically add that missing gene back. So when we look at the Friedreich ataxia, so the underlying cause of the disease was identified in the frataxin gene, and it is a repeat expansion in an intron, which leads to a shutdown of that genetic locus. That means that the patients make very little frataxin. And I think it's an additional plus that we don't have a full loss of function in the patients. We make a little bit of frataxin, which should prevent that we get an immune reaction against the transgene we put in. So in other diseases where like no factor VIII is made at all, and the patient has never seen that protein, then you also get an immune reaction to that basically now new protein to the patient. So in that makes Friedreich ataxia really super attractive for gene therapy. And then in addition, the gene size is quite small, so we didn't have to suffer what was in the Duchenne's to think about engineering how we put a large gene, how we make it smaller and put it into one AAV. So we have really a lot of opportunity. And I think the third point, because frataxin is expressed in all cells, so we actually want to deliver it to like many organs and many cells. So we have a broad choice in terms of the promoter elements, which are very well understood. So don't need to be absolute tissue specific. And we also have, I think, a broad choice on the different capsids. So it has a lot of attractiveness from a science and researcher side, and I think also perspective for getting a good product into the clinic.
So you just talked about the attractiveness of FA due to the many tissue types that can be treated, and that frataxin is in all cells. Can you talk a little bit more about the different tissue types that might be targeted in particular programs?
Yes. So while it's, I said it's a one attractiveness that we feel that there's a benefit even if we treat all the cells, but there's obviously very clear differences how the different tissues are affected by the disease. So from my point of view, the deficiencies in the neuronal cells and the sensory neurons and the cerebellum are a primary target, which from my viewpoint are a must to treat with gene therapy to get really a good benefit for the ataxia. And then the second big tissue is the heart, because of the cardiomyopathy, which is seen so much. But in the design, I think on the programs, so while focusing on these two organs, plus peripheral nervous system and spinal cord, I think it's quite achievable to find one AAV capsid to target heart and brain and periphery, and then have sort of upside that we may cover sufficient of pancreas. So on this, like, we know there's additional issues with the eye. And I think if we want to go, when we look at the experience from the ophthalmology field, that would need a specific administration. I think what the field has learned, we cannot just give eye drops to get the AAVs into the retinal cells. So we would need to do specific injections. And that is, I think, a good discussion in the field. You know, can we come back later to treat, you know, additional parts like the eye, or do we need to do this right from the get-go? But from sort of the research, you know, I'm doing, I think it is to really try to treat where we get the biggest benefit for the patient, and focus on first on those goals, because we cannot do everything at once.
And when a tissue is targeted or a therapy is delivered, is there an estimate or a know of how many cells are expected to be treated to see a therapeutic effect, or what percentage of cells?
Yeah, it's a very complicated question, and I think we have. So and it comes a bit, we go later in the different animal models and cell models we have. So I think for the heart, because we have a very good mouse model to understand about the heart deficiency and the cardiomyopathy, and with that model, we have a good estimate that we believe we need to get a treatment of 50% or half of the cells in the heart. And what we didn't, or I didn't mention before, because frataxin is an intracellular protein, which is basically not secreted. So it's not like an enzyme in a lysosomal storage disease, where you could have the enzyme made in liver, and then it goes around through the rest of the body. It's very then localized. And then in addition, it's not just an intracellular protein, it really goes into the mitochondria and is very important for the mitochondrial health. So like, the more cells we can transduce and target, the better. But we expect to have a clear clinical benefit on the if we get 50% in the heart. For the brain, it is a little bit more difficult to understand. So we would also, because the mouse brain is so much simpler than the human brain. So I think it's like the goal is basically to go to the maximum we can do. In terms of protein expression level, on the other hand, we have really good data from the natural histories and the humans themselves, because we know that the carriers, which are heterozygous, make around 50% of the protein, and there is no disease or phenotype described, in the carriers. So we do have, the good thing is we need to add, plus 40% or plus 50%. So we need to add very little in terms of protein expression. And just to come to the next question, can we add too much? And yes, we have some evidence if we use super strong promoters and do a 20-fold of normal, that at least in the mouse and in the cell cultures, the cardiomyocytes are sensitive and can and it has a toxic effect. So we do have a wide therapeutic window, but as definitely we need to stay inside that therapeutic window to be effective and safe.
So based on the graph that's on the screen,
Chen has a question.
Sorry, go ahead.
Yeah, one of the questions from our audience, I think, is a nice question to ask right now, because they're asking, is there a risk that different cellular types in the body adopt or incorporate the new genetic material at different rates? And if so, what are the implications of that? And I think we were starting to get at that, but this
Yeah.
question.
I think it's not really. I think it's not really a risk. I think it is definitely happening, because when we do the IV injection, we know that the AAVs like to go into the liver, and so in generally, so we have a high, like we can have a higher load of the liver if we look at the AAV8 data, for example. So there's a lot of engineering going on, as Michael said. So we can make new AAVs which don't go as much into the liver, and that's a very achievable goal. So in all the animal studies, and that's why we are doing NHP studies, we look very careful through all the core organs and tissues of the animal model to understand the load per tissue. And basically, a liver toxicity would limit our dose we could for the patient. And I think it's to a later question, that makes all of these programs just long and tedious, because we really need to analyze it in all the different organs and estimate how many vectors go into it and what expression levels do we achieve. But again, the good news is, from the expression level, we have a really wide therapeutic window, and we have a huge range of promoters available, which can assure that we stay in that window. And I think the promoter is more powerful versus having, if you have 2 or 10 particles in the cells. Okay, if you have 1 versus 10, then it's a 10-fold. But 1 versus 2 is a 2-fold. But the promoter differences, they can go 50-fold. So it's like we have sort of a double regulatory system, but we need to always test it.
Got it. I wanted to go back to this graph here.
Yeah.
So we had a question submitted previously asking about dosage. And so would everybody get the same dose, or because everybody has a different range of frataxin in their cells, do you have to do any determination of that frataxin level to decide on dosing?
It's a really excellent question, and it is actually something we are debating also about it. But so I think the first part, there will be a dose adjustment, based on the especially if you give IV, it's always based per kilogram, because obviously a child which is 10 kilogram needs a different dose versus an adult who is 70 kilogram. In terms, but back to the therapeutic window, so the patients, can have anything, and I think we still would like to have more data from the natural history studies around us, and also for more tissues, because most data we have is from blood serum, and then it's like, how much does the blood serum reflect, really our target tissues of the heart and the brain, and there is very little CSF material available, because it is not often collected from Friedreich ataxia patients. So we have then for the brain even less information. But back to this relatively large therapeutic window, we don't think we need to adjust to the patients to the patient level, and especially if we look at blood. How does it correlate then, to the cerebellum? It's like we don't know.
So I wanted to, the question came in too in terms of if the target tissue cannot be measured directly, what kind of tests or assessments are being thought about to be likely performed? We know that patients, we're doing a biomarker study using MRIs. You talked about blood levels of frataxin. Sometimes patients in the clinic have gotten nerve conduction studies. We definitely use the mFARS. is there a way of being able to kind of use the tools that we have to be able to assess that there's a therapeutic effect?
I mean, so I think what especially FARA has done in building up the consortia and getting the natural history studies now going for a long time and adding on to it, and also starting new initiative is absolutely critical for us, and it's like we could really not move forward without all these resources. There is definitely need for more. So in the gene therapy, what we really would love to measure is the transgene expression. So, and then we are a bit like, while it is a good news that we need very little frataxin and it's very low expressed, it's also the bad news. It is very difficult to detect. And there are, many academics also working on novel tools and also in the industry. And that is really, so we still need to get more sensitive methods. And there's multiple efforts, and Helen Puccio, has developed a new antibody, which seems to be superior to what was before, but I think there's still the desire to have even better tools. And then one issue is when we do our non-human primate study, they are very, like in terms of the gene, it's always a good and a bad. It's good that it's so similar to human, but then maybe we want to differentiate between the drug we put in or the gene therapy we add to the endogenous non-human primate, it is very challenging. So I think the TRACK-FA with the imaging, which has just started, is also one of those fantastic tools. I think for the CNS to have more imaging markers will be super helpful. I think Jenny, you wanted to question.
Yeah, we've got a few questions that have come in from the audience that I thought are good to maybe tie together what different concepts that we've been talking about. And so, Michael, maybe I'll throw this one out to you. Do we have an indication of the duration of the therapeutic effect of a new gene delivered to a cell?
Well, it certainly seems with AAV that you can expect, as long as the cell stays alive, you can expect that there will be long-term expression. And there are plenty of examples now in animal systems of expression that is sustained out for over a decade. How much longer? Difficult to know, but it does depend a little bit on the rate of division of the cell that is originally transduced, because the episome, if there's it will be diluted as cells divide. Now, fortunately, in the brain, cell turnover is generally very low, so I think we can reasonably expect long-term expression over many, many years. But obviously, ultimately, the proof will be in clinical testing.
So I guess that just might be another then, right, to FA, and that the tissues we need to target are tissues that don't turn over very much, right? They're in terms of the nervous system and the heart, are my understanding those organs, those cells are not turning over on a regular basis.
Exactly right.
To add on, I mean, like for the neurons, there's data which clearly demonstrate the 15-year, activity, but it just takes time. I think, as we have learned from the hemophilia field, to really get the years down. But I would expect that it should be lifelong for the nervous system, and also very long for the heart.
So the other questions that are coming in, I think, are related to, the concepts that we've talked about, which are, in trying to design a gene therapy program, we have these different variables, right? We have the virus, we have what we put in the virus, not only the gene, but the promoter that that you talked about, Gabby, different types of promoters. We have where we want it to go, what tissue, and what modes of delivery. And so some of the questions now are trying to pull some of these together. And so does a specific AAV to target one cell type preclude using a different AAV later or even simultaneously to target a different part of the body?
So I think this question comes really to the immune response to these vectors. So we'd expect after we deliver a gene therapy that the body will make antibodies in particular, but also develop T cells, a cellular immune response to proteins or peptides derived from the vector capsid. And these types of AAVs were initially characterized because antibodies could discriminate between them, and that's certainly true. There are some members of the family that antibodies to one capsid will not cross-react with another, but there is a fair amount of overlap. So delayed delivery of an AAV has to get over this immune response that effectively can neutralize the vector when it's introduced at a later time. Simultaneous delivery shouldn't really be a problem from an immunological point of view. I don't know whether that gets to the question, but I think.
Yeah, I think
that
was the second half of the question was simultaneous. Yeah.
It's like when there's I think to add on to it. I mean, that's a little further out. I mean, there's a lot of research going on how to suppress the immune response, and to enable either redosing, or also because of me also what we did not touch base on it because we are all exposed to AAVs in our life somewhere. And so we all carry some pre-existing antibodies. So at this point in time, everybody will get screened for the pre-existing load. And that is much lower than after a treatment. But there's definitely, there's a lot of effort, research going into how can we treat patients which have already neutralizing antibodies against a particular AAV. And then there's also research, just like how to prevent some of these strong immune responses. But at this point, I agree with everything Michael said. We
don't have the technology at the moment to be able to do it.
Right. Yeah, and I guess that's Ron Bartek submitted a question, I think, along the same lines of what you've been trying to address, which is, are you able to imagine a time period after which we might expect to consider a second dose of a gene therapy? So is there a time element, but also just an evolution of the field and what we can do medically?
So when I think from the eye field, they can do in the eye, you have a different, because of the compartment, they can dose on different time points. And I think they wanted that also because you don't want to, like if there's any adverse effect, you don't want to lose both eyes. But in terms of systemic, it is or like high doses, it's just at this point, I think it's not possible. But Michael.
So we do see a decline in neutralizing antibodies over time, but it's a fairly long time course. And as Gabby alluded to, there are technologies that either could suppress the immune response at the initial exposure, or that can get rid of the antibodies that might prevent repeat dosing for a short period at some future time. So, Gabby's right. There are technologies in the works that may enable these things, but at the moment, that separation in time is still a problem. And there are many research projects focused on management of these immune responses.
Jen, we're good?
Yeah, I think so. I think that the last question that I think kind of fits in this category, I think we've mostly addressed, but I think it comes down to just the question's asking, would the treatment of all major systems affected be required in order for a gene therapy to move forward? Or should we expect that, there might be a few systems that can be treated, maybe even a few systems or a few areas within the brain that could be treated, and that might be, an effective therapy, but might not get us to a full cure.
Mhm.
You go first, Michael.
Certainly, it's a very complex picture to try and achieve frataxin expression at an appropriate level in multiple tissues all at once. So my feeling is it's likely that we'll initially have to target a group of organs at a time, rather than aim to fix everything at once. So I think that's the direction, but it adds a complexity from a clinical development and regulatory standpoint and so on, to do that.
Yes,
But,
I think a full cure, I think, is far away, especially because of the complexity in the many tissues, and then it's like how you define the full cure. So I think with gene therapy we have to get our expectations right, and I think we all lowered our expectations somewhat, that we want to start from stopping disease progression, then potential reversal of some parts. And I think one point we haven't touched on with gene therapy, we will not bring cells back which have been lost. And part of the disease course is like we really ideally want to treat. So I think our first attempt of the gene therapy is not a preventive gene therapy to treat the patient before there are symptoms, but we would want to treat as early as possible so that we have, enough cells there which can be rescued. Because if a cell is beyond a point and is going to die, then the adding of the frataxin gene will probably not have any benefit. So it is a complex situation in a way. So we need to learn really what gene therapy can deliver.
So based on what we've talked about so far, it is clear for FA that there are many, many considerations for approach, target tissue, AAV choice. But I wanted to also give an overview of the different steps in terms of validating that approach, the approach that's chosen. And so we have a slide here with kind of some different cartoons, but if each of you could speak to the different stages of kind of the validating that proof of concept, and what do you get? Like, what are you looking for when you use cells or move to a mouse model or primate? If you can just review a little bit of the data that's important to the program development.
Yeah, so if I start, and Michael, please wave when you want to chime in. So like what is missing on it, I mean, the first round is really this molecular biology where we design what is the world of those, basically of the payload we can make with the promoters. And there we use a lot of cell lines and also the patient-derived cell lines and iPS cells to really understand the expression levels, the functionality of the transgene, that everything works what it wills. So in terms of the patient-derived cell lines, it's in this case a real critical tool in connecting all the pieces, because we have very limited mouse models for Friedreich ataxia. And they are also not quite the same as a patient, because they're basically missing all of the frataxin protein expression in specific organs, because we cannot like the repeat expansion in the mouse just doesn't show a phenotype. So if the mouse has a different sort of overall network, which they're not so susceptible. And so when we go, we don't have any large animal, and that is a little bit of gap in that field because we cannot model it in the right organ structures and also in a larger size. So we go from the while the mouse is extremely good for efficacy in the heart. So we then need to model how the AAV goes to the different tissues in a non-human primate. And then we use, in addition, the iPS cells, because we can make from the iPS cell the relevant cell types like sensory neurons, cardiomyocytes. And we do see that these cells express less frataxin, that they have like the hallmarks of the disease with the iron transport deficiencies and etc. So we can use these in vitro systems of the patient-derived cells to understand that our gene therapy does work on the critical cell types, and then in the non-human primate we can really map where does the virus go, how much protein do we make, and we do understand the levels of protein we want to reach, and really importantly, do we see any side effects? So like it would be, I think, nicer and better if we could do it in a disease model, because we may not see certain side effects, because in a disease model certain cells may be more susceptible, and that's what we try again to match with the iPS cells, because like we could see if cells die, if we put too much frataxin in and stuff like that. So that, and then basically the dose estimation comes really by using all three models together, and there's a lot of modeling involved, and also then the route of administration. What does intrathecal give us versus intracisterna magna versus IV? That is all done in non-human primate. Maybe Michael, if you have any additions. You're mute. We can't hear you.
When we get to looking at setting up clinical trials, in gene therapy, clinical trials work very differently from any other, because it's a therapy you can give once and you can't, at the moment, we can't repeat it. So we kind of have to get the dose right at the outset. The first trials are all about safety. And, if this is delivering systemically, it might be at a high dose, and we need to be aware of adverse effects that can occur with systemic high dose, or if it's directly into the CSF, then there are different kinds of problems. But the key thing is we have to start at a dose that we think is going to be effective, and that's unlike most other clinical trial settings. And then initial studies studying safety would look for measures that would indicate levels of expression or benefit. And some of the work that FARA has been doing in terms of biomarkers, and we have our own programs in terms of biomarkers, are may be very important in early studies to demonstrate that we're getting some expression and there is some potential benefit. But that's challenging, particularly in the CNS. And then we'd progress into trials to look for evidence of efficacy using endpoints such as maybe imaging or the FARA scales, wearables, and all the rest of it. But the initial studies are going to be very critical to have endpoints that we can measure, get some confidence in the levels of expression and safety and early evidence of potential clinical benefit.
Thank you. We are coming up on the hour. Jen, any final questions that we want to
Yeah, we have a few final questions, but I think a few of them you've started to address that, kind of how we use the information we get from these preclinical models to think about designing, that first in human study, and what those escalating doses look like. And one question is, would those need to vary based on a patient's or an individual's genetic burden in terms of the length of their GA repeat? And I think that might also go to what we were talking about before. Would a starting dose be based on how much frataxin someone starts with to begin with? And I think what Gabby mentioned earlier is probably not, right? That initial.
No, probably not. I mean, we said we cannot disclose specific designs, but
Yeah.
I mean, there's always very clear, inclusion criteria for the phase 1. But in terms of, we know that the GAA length is an indicator, but it's not an absolute predictor of the how the disease progresses, and also for the severe diseases, the severity. But it's like, again, the phase 1, it will be a quite small trial. It has to be an effective dose, because as I said, we cannot redose. It also has to be very safe, because we also cannot turn it off. So I think it's very different in the gene therapy. And when you look at the other trials, they also go very slow. That will be one patient dose, and there will be a waiting period to understand that there is nothing unpredictable to happen. But I don't think that GAA length or frataxin protein level will be that major criteria.
And just one of maybe this is an interesting point to end on for next year's symposium, but one of the comments was, are there research efforts, is industry looking at non-viral delivery methods as a way to avoid the immune response, and to be able to deliver therapies multiple times or to other organ tissues?
Yes, we do, but it is a very early field, especially for CNS. So I mean, I think we all keep. For Takeda, we always look at innovation. We look at we're very open to new approaches. It's like we also cannot abandon constantly efforts. Otherwise, we never bring anything into the clinic. And it is so it's like with a lot of the new technologies, including gene editing, etc. They have different issues which we don't know yet. So I think it's a mixture. We need to do things which we understand the risk, and we understand, at least because gene therapy is so new, so there's a lot of stuff we don't know. So we need to do a careful approach, and the non-viral is just not here yet. I think it's for CNS probably a decade away from what I'm seeing.
So for FA, there is an element to be appreciative of all that is being learned in other diseases that could guide development and design, for safe and effective trials for FA. So it's no knowledge is lost or goes to waste. Thank you so much. I've learned a lot in this session, and I know I've thought of some additional questions. So we are going to end the session for now. We have a session again later on, starting at 7 p.m. Eastern time. So please join us. We'll be talking more about the specifics of clinical trial designs and what that looks like compared to what we know for traditional drug trials. But if you do have additional questions, please go ahead and send them to info@curefa.org. We can reach out to our panelists, and we'll try to get you as many answers as we can. Thank you very much for joining us, and we look forward to seeing you again later on today and have a good rest of the afternoon.
Michael and Gabby,
you very
thank you
much.
so much.
Yeah, thank you.
Thanks
Yeah,
so
thank you
much.
for having us. Thank you. Happy to be here.